everyone! I thought you might want to see me, just get a glimpse before I start talking about photosynthesis. And I also thought maybe I'd introduce one of my cats too. This is Oli. She's a little tiny kitty. If you hear anyone crying in the background, it's probably her. Say hi. Say hi to everybody. Okay, I'll let you go. I gotta go talk about photosynthesis now. Okay. So let's get into it and let's talk about photosynthesis, at least introduce you to the topic. So let's do a little review before we start getting into a lot of the details of photosynthesis. Photosynthesis is the process by which the plant cells are going to use light and carbon dioxide to make glucose and energy for the plant. And in addition to that, the plants are going to release oxygen. Because the plants will be able to use this process to make their own food from carbon dioxide and light energy, plants are referred to as autotrophic. Hopefully this is review for you from Gen Bio 1. And then also remember that animals will require other organisms to provide their food, and therefore they're considered heterotrophic. So plants are autotrophic, and some other bacteria that we're going to talk about, and animals are heterotrophic. The transfer of energy from one form to another is the basic nature of the process of photosynthesis. And the key to the whole process is carbon fixation. But before the plant can make the glucose from carbon dioxide, that energy has to be transformed into chemical energy, which will then be used by the cell for what's called carbon fixation. Yeti, please stop crying. Okay. The process of transferring energy and using the energy is going to entail two steps in the process. One which will use light, and the other which does not directly require the light. So let's talk about those now. Yes, you're very excited, aren't you, buddy? I don't know if you can hear that, but Yeti, my other cat, is crying right now. So let's talk about light-dependent and light-independent reactions. Light-dependent reactions, as the name suggests, these are reactions that need light to work. They capture energy from the light, and they use special pigment molecules that collect the energy and collect the energy from the light and transform that energy into a chemical form. This transformation that happens, it consists of making ATP from this energy and reducing NADP, which is going to be used in the synthesis reaction. So we'll talk about that in a second. If you think back to when you learned about glycolysis in Gen Bio 1 and some of the other processes you learned about, you'll remember that there was the action of passing electrons along a series of proteins to enable cells to make ATP. Really, the action of the light reactions is pretty much the same. And if you are having trouble remembering those, you should review that section of the book in your notes to make sure you're able to understand the connections I'm going to point out in a little bit. So that's talking about the light-dependent reactions. Now let's talk about the light-independent or the dark reactions. Now these don't have to occur in the dark, but they just don't directly require the presence of light for these reactions to occur. And in these reactions, as you'll see, the main part of this is the Calvin cycle, and the ATP and the NADPH, which are formed in light reactions, will be used by the cell in the process of carbon fixation. So when we use this word, carbon fixation, we don't mean that the carbon's broken. What we mean is that we will take carbon in the form of carbon dioxide and use this in the synthesis of sugars and other molecules. So it's not being broken. What it means, and I'll say that again, what we mean by carbon fixation is that we'll take carbon in the form of carbon dioxide, CO2, and use this in the synthesis of sugars and other molecules. That's what we mean by carbon fixation. And as you should see, even though these two are referred to as different reactions, the light dependent and the light independent, they're not really independent of each other. The dark reactions depend on the formation of ATP and NADPH in the light reactions to provide the energy necessary to carry out the carbon fixation reactions. One of the important quote-unquote waste products of the process of photosynthesis is oxygen. But when we consider the reactions, you'll notice the oxygen is not from carbon dioxide in the system, but it's going to come from the process of breaking water. And that's a really important point I'm going to make when we actually get to the reactions, is that this waste product of oxygen is not coming from carbon dioxide, but it's coming from the breaking of water molecules. So what do we mean by the process of carbon fixation, and how does this tell us where the oxygen comes from? So in the process of photosynthesis, one molecule of carbon dioxide and water will be used in the presence of light energy to produce a molecule of carbohydrate and release a molecule of oxygen. But plants aren't the only autotrophic organisms on Earth. There are certain types of bacteria which are able to make their own carbohydrates from carbon dioxide, and these are referred to as purple sulfur bacteria. Take one guess at what color they are. They're purple. And they stinky because, as you can see, the main difference between these reactions is that they are using hydrogen sulfide instead of water to fuel their reactions. And then what you end up with is sulfur at the end of the reaction. So they're real stinky. They stink, if that helps you remember them at all. And so in the case of these purple sulfur bacteria, it's not the carbon dioxide in the H2O, but it's carbon dioxide and hydrogen sulfide, which they'll then use to form CH2O, which is also formaldehyde. So you get formation of formaldehyde in both of these reactions. And so what you can see in both of these reactions is that the oxygen that's in the carbon dioxide isn't released, like I said before. It's the oxygen that's in the water that's released during photosynthesis. So I'm just reiterating that point again, is that the oxygen that's being released is from the water and not from the carbon dioxide. If you look at both the bacteria and the plants, you should notice a general equation that you can use to summarize the process. That's what's right here. And in this case, we'll use an A to designate the S or the O, depending on which of the processes we're referring to. So this is just a general equation that represents the equation for sulfur bacteria and for photosynthesis. And so this A can represent multiple different molecules. This is going to tell us that one molecule will be an electron donor and one of the molecules will be an electron acceptor. And what do we call reactions that will involve the transfer of electrons? We call them redox. Or oxidation reduction reactions. Really doubt Brother Albert did that in his video. Anyway, if a molecule gains an electron, it will be reduced. If a molecule loses an electron, it'll become oxidized. I'll say that again. So when we're talking about redox, it's when a molecule gains an electron, it's reduced. If a molecule loses an electron, it will become oxidized. So in the case of our reaction here, the transfer of electrons will be shown by the presence of hydrogen atoms on the molecule, and the more hydrogen atoms on a molecule, the more reduced it will be. Okay. So, Let me make sure. Okay, so we're going to stay on this slide. So from this quick review of redox reactions, which of the molecules in the general reaction is oxidized and which is reduced? Well, the H2A in this general equation is oxidized because the loss of the H means that there's the loss of electrons by that molecule. The CO2 increases the number of hydrogens, and therefore the number of electrons, and therefore the CO2 is reduced. I'll say that again so that you get it down in your notes. So the H2A is oxidized because when it loses that H, it means there's a loss of electrons by that molecule. And the CO2, the carbon dioxide, increases the number of hydrogens, and therefore the number of electrons. So the CO2 molecule is reduced. Another way that you can state that is because the H2A is losing electrons. This will be called the electron donor. So because this molecule here, the H2A, is losing electrons, it'll be called the electron donor. And because the CO2 is gaining electrons, you can see it's got this gain of electrons here, it's going to be called the electron acceptor. So we call this one the electron donor, this one the electron acceptor, and it's all based around whether it's losing or gaining hydrogen molecules. So back to photosynthesis, what this means is that in the process of photosynthesis, the water, the H2O, is the electron donor. It will be oxidized to O2, and the CO2 is the electron acceptor, and it will be reduced to CH2O, or formaldehyde. This overall process should look familiar to you, hopefully. Where have you maybe already seen this similar process? You've seen it in glycolysis, you've seen it in the Krebs cycle, you've seen it in the electron transport chain, and in this case here, the glucose molecule will be oxidized to CO2. O2 will act as a terminal electron acceptor, which will be reduced to H2O. And this is an important comparison to make because, as you're going to see, the light-dependent reactions can kind of be seen as a reversal of the electron transport chain. And that's because many of the same processes are involved, and the process of the dark reaction will be a reversal of glycolysis. Of course, what I've covered right here is an oversimplification of the reactions, but you should be able to see how these processes are the reverse of each other here. So like animal cells, the primary sugar which will be used by the plant cell will be glucose. As a reminder, glucose is a 6-carbon sugar that'll be used for many of the processes in the cell. Therefore, for photosynthesis, we can summarize the process as 6 carbon dioxide molecules that are combined with 12 water molecules, and it uses light energy to form 1 molecule of glucose and release 6 molecules of oxygen gas. So I'll say that again as a summary of what is going on in photosynthesis. So we can summarize the process of photosynthesis as 6 carbon dioxide molecules, CO2 molecules, are combined with 12 water molecules, 12 H2O molecules, and photosynthesis uses light energy to form 1 molecule of glucose and release 6 molecules of oxygen gas. And so I just wanted to talk today, or in this video, about carbon fixation, the basic reactions that are happening in photosynthesis and in other autotrophs like purple sulfur bacteria. And then in my next video, I'll talk about where photosynthesis takes place and what the structure of chlorophyll and chloroplast looks like. Okay, see you soon. BRB.